All right, thank you very much for the kind introduction and invitation to speak today. So I'm going to kind of give you a glimpse of what my group works on. And so my group kind of thinks about microbial communities maybe a little bit differently than some of the people in the room. So I'm interested in thinking about them as agents for bioprocessing or other types of applications. And so in general, I think about how can I divide labor amongst different microbes in a community in order to carry out some sort of complex tasks. And so why do I want to do that? So we're interested in approaching these problems in a way that we can reduce metabolic load on individual cells. So as biological engineers, we often try to make E. coli, or our favorite bug of interest, do one more task and one more task, and then we find that they grow poorer and poorer because of the metabolic burden or other burden on the cells. And so if we can divide a task amongst more organisms, can you have everyone grow faster, have higher fitness, and then make something different? The other advantage, a key advantage, is taking advantage of the capabilities of different types of organisms. And so as much as we love, our Saccharomyces cerevisiae and our E. coli, they can't do all the kinds of chemistries we're interested in. We've learned a lot about different chemistries this weekend, or this week. It's spring break. I don't know what day of the week it is anymore. And there's beautiful chemistries like being able to break down toluene and benzene that pseudomonads can do that we can't get to work in E. coli or other types of organisms. And then, of course, as someone coming from the early synthetic biology realm, the idea of modularity is really exciting. And so, I started out thinking about the modules in terms of promoters and proteins and switching gene expression on and off during different types of conditions. And I will tell you a little bit about what we work on there. But I'm excited about thinking about microbes as modules for different types of applications. And so, can I one day have some sort of cellulosic biomass and assemble a microbial community that will grow and function in the way I want and make a biofuel? And the next day, do that same process, swap out a couple microbes, and make an antibiotic. And can we have this happen using typical types of bioprocessing equipment, have it be flexible, modular, work with available downstream processing components in a quick and effective way? And so in order to do that, there's lots of fun, exciting challenges, and hopefully my group is working on a lot of those, and so I'll tell you a little bit about what we're working on. And so in order to kind of think about how you're going to get these organisms to work together, we can think about essentially getting them to coordinate their behavior, and so we can address that by thinking about cell-cell communication. We can think about manipulating community composition. We've talked about organisms we can't grow in the lab, but we also have a problem where you often try to grow multiple organisms together in a culture, and you have one out competing the rest of them. And so we have those kinds of challenges as well. And then, of course, reaction pathway engineering. How do we think about dividing processes amongst organisms in a way that leads to the modularity that we want and the production of the compounds we want? And so I'm going to start by telling you about what my group has done towards engineering cell-cell communication. That's kind of where I started my career. And so to address cell-cell communication, we take advantage of essentially quorum sensing systems. So these systems that have evolved in bacteria in order to sense and respond to changes in their local gene expression. And these systems are composed of essentially the production of a small molecule and the sensing of a small molecule, and we can essentially decompose those components and rewire them in different types of ways. And so the target that my group started with was, can we use these systems to engineer interspecies communication? And so lots of early synthetic biology used these components, these quorum sensing components, in order to enable things like predator-prey ecosystems, or making of things that mimicked pattern formation. But essentially, it was often E. coli to E. coli communication, making these cool model systems. And so I started my career. I wanted to make something where it was a little bit more likely to make it to bioprocessing in the real world during my career lifetime. And also, I wanted to make it so that we could communicate beyond E. coli and E. coli, or E. coli and Pseudomonas, or some kind of normal organism that had these types of communication modules. And so we wanted to think about, can we rewire existing interspecies communication systems to really enable interspecies? And I wanted to go beyond gram negative to gram negative and say, well, let's go gram negative to gram positive. We don't know available communication systems that do this. The AI2 system, of course, is able to do intra or interspecies, but that's one communication signal. You're not going to be building, right, interspecies communication in a really nice way using those systems. And so our targets were communication between E. coli, because everyone loves E. coli, at least us bioengineers do. So it's gram-negative, it's a great cloning host. It makes a lot of recombinant protein, and it's a workhorse of bioprocessing for small molecules, as Christilla told us about a while ago as well. And then Bacillus megaterium. And so we wanted to target a gram-positive organism, and we chose this organism because it stably maintains plasmids. At the time was a little bit easier to work with than some of the other Bacillus strains. It's used in industry for different types of bioprocesses, including vitamin production. It's generally regarded as safe as well. And interestingly, it has low extracellular protease activity, and it's a pretty good protein secretion organism. And I'm going to tell you when I get to our applications realm how we're taking advantage of that particular property. And so if I take these two organisms and I tell you I want to make them talk to each other, right, how are we going to go about that? So we're going to take advantage of natural communication systems. And so my group started working with the ESA-R quorum sensing system. And so it's interesting. It was interesting to us because unlike LuxR, which is described at the top here, where essentially you have, in the absence of a signal molecule, the LuxR protein is unfolded, and RNA polymerase can't activate gene expression at the LuxR promoter. Once you add the signal molecule, which is this acyl homoserine lactone, you see LuxR folds, it recruits RNA polymerase, and you turn on gene expression. And so this is not a very modular setup, but it works pretty nicely. Whereas we have ESA-R, right, recognizes the same small molecule, so the same 3-oxohexanyl homoserine lactone, but it acts much more like LAC-I, right, or the ERA system. And so in the absence of signal molecule, it sits down on this promoter, blocks gene expression, RNA polymerase can't get to it. Then you add the signal molecule, it releases, and you see gene expression turning on. And so we were excited about this system. It has different dynamics. We thought we could make different types of promoter type systems, which we've also done, so AND gate type systems. But one of the things that we thought was really cool about it was that if you swap out this promoter, so you move from this ESA-I promoter to the ESA-S promoter, then essentially the ESA-R becomes an activator, and gene expression is now on in the absence of signal molecule, and you can add the signal molecule and turn it off. And so there are, of course, many examples of promoter systems like this in nature, but there's not a lot of examples like this that are available for biotechnology. And so we were really excited about taking advantage of this system, engineering it, and having it work for different types of application, where you could add a single signal molecule and turn some genes on and some genes off, which can be really important for metabolic engineering applications, especially when you want to switch from the cells growing to production or other types of dynamic processes. But one of the things we found was this wild-type ESA-R required very high concentrations of signal molecule in order to turn on gene expression. And so we thought that was a big challenge, and we wanted to overcome it. And we also, like most synthetic biologists, want to have a lot of tools in our toolbox. And so to do this, we used a directed evolution approach to make essentially a set of ESA-R variants. And so we used a dual on and off screening system, where essentially we made a large library using error-prone PCR, kind of traditional approach, screened that library for essentially the ability to repress gene expression. We used a luciferase reporter, so any dark colonies we picked. And that was to make sure that our pool of proteins were all functional proteins. The next thing we did was we took all those functional proteins or functional variants and screened them for increased sensitivity to that small molecule, to our 3OC6HSL. And this is essentially a summary of our best results. And so the black over here is the wild type. Our best variant is this D91G, which showed a 3-fold increase, or 300-fold increase in signal sensitivity in this expression assay. And we also have intermediates that are about halfway in between. And then one of our variants had a couple of mutations. And one of them actually still has an active ESA-R, but it requires 10-fold higher concentrations. And so in a single round of directed evolution, we now have a suite of ESA-R variants that function over about 4 orders of magnitude. And so these kinds of tools now allow us to have switching right, at exactly the conditions, right, cell density or small molecule addition that are important for whatever application you're using. And as I mentioned, you can swap out the promoter, so switching from the ESA-R protein to the promoter to the ESA-S protein, and you essentially can have gene expression turning off instead of turning on as a function of small molecule addition. All right, so far I've told you about my grand challenge of making E. coli and Bacillus talk to each other, right? And this is all I've shown you. So, you know, E. coli responds to acetylmyrcene lactones, and most of you probably knew that already because you've either used an assay, read a paper, or something about that. But the ESA-R system is still pretty cool. But what about the rest of this? So our first task was, let's see if we can get Bacillus to produce acetylmyrcene lactone. So we know what proteins are involved. So we express this ESA-I protein. In Bacillus megaterium, it's a xylose-inducible T7 type of system. And we know that ESA-I, from lots of different types of characterization, requires S-adenosylmethionine and 3-oxohexanoyl acyl carrier protein. So 2 easy substrates. Hopefully it works. Our initial experiments showed us we didn't get any product. And of course, when you don't get any product, it can be, right, myriad problems. But my grad student, Raphael, at the time, did some research, looked into the organisms we're working with, and noted that the Bacillus likes to produce a lot of branched chain fatty acids. And so that indicated that maybe we didn't have enough of this particular substrate. Maybe there was only essentially branched chain fatty acids, and we were either making some variant acetylmyrcene lactone with branched chains, or we just weren't making any because there wasn't enough substrate in that system. What we also identified was that previous research had identified some conditions that allowed Bacillus to make more straight chain fatty acids. And so we tried a couple of those conditions. The first one we tried was low temperature, and then we also tried minimal media, so a defined media instead of LB or some typical, laboratory media. And those both worked very well. I'm showing you here the minimal media system. And so this is a luminescent reporter system in E. coli. And so this is in the absence of acetylmyrcene lactone, and this is with 100 nanomolar. And so here I'm showing you supernatants from Bacillus megaterium, either with an empty T7 vector or the T7 vector expressing ESA-I. And you can see now the supernatant from these cells does contain 3-OC6HSL, and we are seeing nice expression of the signal molecule. It did work quite well as well with the low temperature, but for real bioprocessing applications, we don't want to be having to run our experiments at 22 Celsius. And so then our next step was to actually see if we could do this with some communication between the cells. So we co-cultured our Bacillus and our E. coli. In this case, we used an M-cherry producing E. coli that responds to the 3-OC6HSL. Because of the size difference of these cells, they actually sediment a little bit differently when we centrifuge them. So you can actually see the E. coli and the Bacillus here. Right? This is a microcentrifuge tube, not candy, which you guys probably didn't necessarily visualize it like that because you just had snacks. And so then we can, of course, quantify this. So here we're looking at the fluorescence relative to essentially cell population. Our controls here are on the left, where we have essentially our 0 and then titrating amounts of 3-OC6HSL, our control cells that don't have any ESA-I in them, and then essentially the co-cultures are here. Where essentially what we're doing is we're keeping our starting concentration of Bacillus the same, and then we're titrating the amount of E. coli. The gray bars are uninduced, and the black ones are induced. And so we can see nice communication right through especially 1-to-1 and 10-to-1 ratios, and broader when we do induce a system. The only time it doesn't really work is up here at the very high concentrations of E. coli. And I wouldn't be surprising any of you when I said that when we have those high concentrations of E. coli, it just outcompetes the Bacillus, and we don't have any communication happening in those systems. And so now, right, we've got a system where we have E. coli and Bacillus talking to each other, right, where Bacillus sends a signal and E. coli receives it. What about the other direction? So to address this challenge, we decided we wanted to bring in a completely different quorum sensing system that synthetic biologists hadn't started to use yet. And that was peptide-based quorum sensing systems, and these are prevalent in gram-positive organisms. We wanted to target this both because we wanted a new kind of communication system, and also because we wanted essentially communication lines, chemical lines between cells that would have minimal crosstalk. And so this is the quorum sensing system from Staphylococcus aureus. In this case, you have a pretty similar architecture where you produce a small molecule, and then once it accumulates to high levels, it's recognized, and you have essentially changes in gene expression or changes in phenotype. We have the CA components, which make this two-component regulatory system, and the B and D components, which together essentially process this propeptide and make this peptide AIP. And this is your signal molecule here. And so the first thing that was of interest was proving that E. coli could make the, this AIP protein. Luckily, Alexander Horswell and his group had actually shown some evidence that this was actually possible. And we were able to verify it. So essentially, we expressed AGR B and D in a topovector from E. coli, induced at very low levels. And you can see when we used a Staph aureus reporter, right, so essentially this is a reporter system that naturally, or that has been engineered to respond and fluoresce in response to the AIP1, that our media controls and our uninduced supernatants from E. coli don't give us any signal, where we have good fluorescence when we use the supernatants from these E. coli that have expressed the AGR B and D genes. And so we're getting there. So now we have E. coli that can produce this AIP peptide. So what about getting AIP detection in Bacillus megaterium? And so we were cautiously optimistic that this would work. You know, Staph aureus and B megaterium are closer to each other than E. coli anyway. And the two-component regulatory systems we were hoping that they would work. Of course, I'm telling you about it, so punchline given away. Anyway, so our strategy was to express these AGR A and C proteins from Bacillus, and also include this P3 promoter with a GFP reporter downstream of it. And so you'll notice that this graph here looks pretty similar to the Staph aureus reporter one that I just showed you. That's because it pretty much gives you the same result. So when we have these AGR-CA expressing cells, these AIP receivers, they actually respond to the supernatant from E. coli that is producing this AIP1. And we see a really nice increase in gene expression. And you'll note that the fold change in fluorescence is actually quite a bit larger than it was in those Staph aureus reporter cells. And so we now have essentially really sensitive and very nice reporter cells for this system. And of course, we can put this together. So I can co-culture my E. coli and my Bacillus, where I have essentially signal senders on the E. coli side now, and Bacillus on the receiver side. And you can see really nice communication, once again, which is these larger bars in the middle, where I'm titrating the E. coli concentration and keeping the Bacillus concentration stable in these batch cultures. And once again, it's about 1:1 to 10:1 inoculation ratios that give us the best levels of communication. And here I'm just showing that in this system we actually have, in these batch type of systems, our final cell densities of the E. coli and the Bacillus in these good ranges of communication are pretty consistent. So we have minimal resource competition in these particular systems, the way we set them up. Of course, those things will need to get addressed as we move forward. And so we now have a nice platform for two-way communication. We can put all these things together and start to produce different molecules and think about adding different types of responses to small molecules that are important for particular metabolic processes. And so with that, I'm going to transition to what we've been doing towards community composition. And so there's been a lot of kind of interesting work coming out kind of in microbial ecology, and also within the realm of synthetic biology, that's indicated that the cross-feeding or metabolic complementation that we see between auxotrophs works much better than we thought and seems to be much more prevalent in terms of getting organisms to coexist together. And so we wanted to see if we could take advantage of that and essentially push it a little bit further than the E. coli, yeast kind of pairs, and a couple of other pairs that are out there. And so, of course, apply this to our E. coli and Bacillus combination. And so we took auxotrophic pairs of E. coli. So we just got those from the KO collection, so you can have a large suite of organisms or E. coli strains that can't make a particular amino acid. And we paired that with an available leucine auxotroph of bacillus. We've recently gone beyond this, but for this we have the 1 bacillus auxotroph. And so we can essentially put these together in pairs. And I'm starting out here just showing you this on solid phase, because I think from an image perspective, it's a really nice way to see it. So what we've done is we've spot plated essentially E. coli across the top, bacillus across the bottom, and then we co-culture them in the middle. And we've done this for our isoleucine auxotroph of E. coli, our tyrosine, and an arginine auxotroph here. And in each of these 3 cases, you can see that on their own, right, we see essentially no growth, but we put them together and we see nice cross-feeding and metabolic complementation. And so this indicates that this might be a good strategy for getting these 2 organisms to grow together in culture. So what happens if we put them in culture? So I realize this is a pretty dense slide, but hopefully we can kind of get to it kind of looking at the pattern. So in each of these little squares, I have an E. coli auxotroph, which is indicated by the up here, and I have our bacillus auxotroph for leucine. On the outside in orange, I have how the E. coli monoculture grows over a period of 96 hours, so 24-hour time points. And then in the middle, I have the E. coli growth in co-culture. Then we have the bacillus growth in co-culture here, and on the outside, we have the bacillus monoculture. And so in this top 4 across, you can see that in E. coli in monoculture and the bacillus in monoculture, right, they don't grow, or with the bacillus, you actually see a decline in essentially cell mass over that time, or viable cells, cell mass. Whereas essentially the combined cultures, the co-cultures actually grow quite nicely. So we are seeing that metabolic complementation we hope to see for our biomass production. We're looking at some other ones. You can see some tiny bit of growth by the E. coli and bacillus, but certainly not at the levels here. And then in some cases, we see essentially no cross-feeding. One of the interesting things that we found is the pairs that do or don't work are not necessarily the ones that are found if you look at pairs of E. coli cross-feeding. And so this is something we're going to be following up on, looking at transcriptomics and how these organisms are responding to the co-culture environment, as well as looking at the availability of amino acids in supernatants and whether those change in response to co-culture growth, or whether or not it's just kind of a stable system where the cells are always producing a little bit of that background amino acid. We're also expanding this, of course, to looking at other auxotrophs of bacillus. And so this is really nice looking at batch culture, so looking at small flask growth. But what about continuous culture? So what I'm showing you here is from our essentially approximately 20 mil chemostats that my student Aaron built in our lab. And so essentially we use a peristaltic pump and allow our cells to grow, and we can currently run about 20 of these, and we're expanding to be able to run about 40 of these mini chemostats in parallel, so we can start to ask questions about how the monocultures grow and how the co-cultures grow. And interestingly enough, we see that three of the 4 that worked really nicely in our batch also work well in the cross-feeding. So our monocultures are here, and they wash out really quickly. But in co-culture, the E. coli and the bacillus, once again represented by blue and orange, which I should indicate better on this slide, grow really nicely in co-culture. Our one exception here is this phenylalanine, which grew really nicely in batch, but doesn't seem to grow very well in continuous, and we've repeated this over and over. And so we have new questions of mechanisms that we can look into regarding the cross-feeding in these types of systems. All right, so I can tell you a little bit about our reaction pathway engineering. And so our model process is essentially going from cellulosic biomass to whatever target we want, which I know is not a foreign concept to many of you in this room. The reason we love bacillus is because it's good at secreting proteins. So unlike E. coli, which is not very good at secreting proteins, as much as some biological engineers like to try to make it secrete proteins, that periplasm is kind of a problem for getting high yields of protein secretion. Right? Gram-positive organisms can deal with this differently. And we've been working to engineer bacillus to secrete large amounts of protein. And then, of course, E. coli can be our workhorse, can take up those freed sugars and produce target molecule of interest, whether or not this is a fuel, an anti-cancer compound, et cetera. And so what we want to do is develop a cold culture system that will degrade cellulose and generate target products. So right now we've been working to essentially secrete two different cellulases, this EGI1, which goes from cellulose to cellodextrin, and then also a second bacillus cellulase, which can go all the way from cellulose down to glucose. And so one of the cool things that we found was that we could secrete them. And so we've secreted both of these proteins by essentially modifying a signal secretion system. So we essentially take the signal peptide, and then we include a linker, and then have our protein. And that seems to be the trick in terms of making this modular. There are interesting secondary structure requirements on both sides of the signal peptide. And this actually seems to address those. If you're trying to make a biologic, this is not going to work because you're essentially adding some extra amino acids at the end terminus of your protein. But if you are making an enzyme that doesn't seem to care about a few extra amino acids at the end, it works really nicely. And we tried some different types of tags on here, and you can see looking at both our essentially secretion information on the SDS page, and then also protein activity that these work pretty well. So the cool thing that we found is that there's some great synergy between these two proteins. And so what I'm showing you here is at 30 and 37, what happens if we have total of 5 mg per liter of protein. This is a really low concentration of protein, and we're seeing pretty good cellulase activity. Of course, we'll want to ramp up protein expression as we go, but the cool thing is the synergy. And so what I'm showing you here is across each of these bars, we have a total of 5 mg per liter, but we're changing the ratio of these two enzymes, essentially from 0 to 10, or 10 to 0, where it's all 5 mgs, and then 1 to 1 in the middle. And so we see the highest activity when you have quite a bit of both proteins, and then this titrates based on, at the different pHs, based on the relative essentially activities of these proteins under those different conditions. And I won't get into the biochemistry of those. But essentially, we're seeing really nice synergy between these organisms, or between these proteins, which means now we can think about co-culturing a few strains of bacillus together that are making different cellulases, and then combine that with E. coli. And we're doing some work on the E. coli side of things as well that hopefully I will get a chance to talk to you about at another meeting. So I just want to end essentially by telling you that if we just take this cell 9A and we use it to essentially degrade some cellulose, we can take that cellulose and now support the growth of E. coli. And so this is E. coli growth. It's kind of a long-term growth right now, but only when we have the cell 9AT and this cellulose substrate can we actually see growth. And so we're moving towards a point where we can actually go from cellulosic biomass to essentially supporting the growth of Bacillus and E. coli, and then having E. coli start to produce our products of interest. We're probably a little bit a ways away from that, but we're moving there in the long term. Now I'm going to skip a bunch of slides because that's the way it works. And essentially go back and tell you my conclusions. So I only got to this first part. I was hoping to get to some of the second part where we're doing some microbiome in the built environment, but I talked too slowly at the beginning, and that's the way it works sometimes. And so I've showed you about some new tools for engineering cell communication, that we can use communication cross-feeding for dividing up, for coordinating growth, and how we're moving towards essentially bioprocessing applications using mixtures of microbes. And if the other part looks cool to you as you're perusing it, come and talk to me later. And so I want to acknowledge JGI for the invitation to talk today, and NSF for supporting most of the work. Thank you very much.